Hi everyone, welcome to the second session of this series. Um, yeah, so we will be talking about carbohydrates, specifically monosaccharides. All right, so let's remind ourselves of the ways to classify monosaccharides. We basically look at its two different properties. The first property is the length of the carbon chain. So, for example, if you have 3 carbons in this monosaccharide, it's called a triose. If you have 5, it's called a pentose. If you have 6, it's called a hexose, and so on, so forth. Or we can also look at its the functional group that it has. If it has an aldehyde, meaning a carbonyl carbon. So a carbonyl carbon, first of all, is a carbon that's double bonded with an oxygen. So an aldehyde is a carbonyl carbon attached to a carbon and a hydrogen, while a ketone is a carbonyl carbon attached to 2 carbons. All right, so this is the key difference between ketone groups and aldehyde groups. And turns out that monosaccharides, they can have either aldehyde groups or ketone group. So yeah, if you have a monosaccharide that has a ketone group, we can call it a ketose, and if it happens to be 3 carbons long, then we call it a triose. Excuse me, sorry. And then if you have a monosaccharide that has an aldehyde group, then we will call it an aldose. And if it happens to have 3 carbons in its carbon chain, we can call it an aldotriose. So basically, this name is one that encompasses both properties of monosaccharides. All right, so take a moment to pause and think about, the name in this format for glucose as well as fructose. All right, hopefully you've got it. Let's look at the answer. So glucose has an aldehyde group and it has 6 carbons in its carbon chain, therefore it's called an aldohexose, while fructose has a carbonyl group and it also has 6 carbons in its carbon chain, and therefore it's called a ketohexose. Moving on, a very important concept is that carbohydrates can have multiple chirocarbons. So what does chirocarbons even mean? Well, if a carbon is connected to 4 different things, we call it asymmetric, and therefore it is chiral because looking at its mirror image, so its compound that's in the mirror, this compound, the mirror compound, won't be able to superimpose, or in other words, overlap with the original compound in 3D space. You can try that yourself. The geometry of this this molecule is a tetrahedral shape. So therefore, if you have 4 different things there and you have a mirror image, the mirror image won't be able to just superimpose on top of the original image. That's just sort of like geometry. Yeah, you can try that yourself. And imagine it rotating in your head and trying to align these different balls together. You'll see that even though you can align, like you can overlap the tetrahedral on this tetrahedral, the colors of the balls won't match. So yeah, and then we call this pair enantiomers. So they are the same compound, but they are different isomers of each other. So we can have L isomer, which means level in Latin, and that means left. And D isomer, which comes from dextro in Latin, and that means right. Why? Because if you look at your hands, you'll realize that your left hands and right hands are enantiomers of each other, or they are like chiral, because your left hand won't be able to superimpose on top of your right hand, right? Because if you try to fit them together, you'll find out that your palm of one of your hands is going to face up, while the palm of your other hand is going to face down. So there's no way that they can superimpose to each other, unless you have a very serious wrist injury or like a twist, which is like, I hope you never have that. So yeah, hope that it always, keeps its chirality. Okay, let's move on. So it turns out that this molecule that we've been examining this whole time is glyceraldehyde. So it's here, right? It is the a very simple sugar, meaning that it only has 3 carbons, and only the central carbon is chiral. Why? Because, as I said before, an asymmetric carbon has to connect to 4 different groups. So if we look at the carbon here highlighting yellow, on top of the central carbon, it is connected to only 3 groups, so it can't be chiral. And the carbon below that's highlighted in blue is connected to 4 groups, yes, though it is not asymmetrical because it is connected to 2 same groups, namely hydrogen atoms. So chemists need to find out a way to represent the chirality of compounds and, the isomers, the L and D isomers. So, but how can you do that on a 2D space without being too, like flamboyant in your drawing? Well, they decided to adopt this thing called perspective formulas. So you can have a carbon with 2 of its groups having wedges. Wedges meaning that it's a single bond that points toward us, away from the plane of this illustration. And it can also it should also have 2 dashes. Dashes meaning that this single bond connecting between these 2 carbons will be pointing away from us, away from the plane of the paper or the screen that's depicting this molecule. Yeah, so it turns out that when the alcohol group's on the right side of the perspective formula, the glyceraldehyde is a D isomer, and when it's on the left side, it's an L isomer. But that's just the way it is. That doesn't mean that, for any organic compound, like if it's drawn in perspective formulas, it's definitely a D isomer. It doesn't really guarantee that. So it just happens to be it this way. All right, so what if we want to be lazier? Well, we can just get rid of the dashes and the wedges and just have our sweet, thin lines back, thin and even lines back. So yeah, this is called Fisher projection formulas, and this, due to the absence of the wedges and the dashes, we heavily rely on the position of the alcohol to determine which isomer it is. So as you can see here, it mirrors or like parallels the isomers here in the perspective formulas, right? For the D isomer, it has the OH on the right drawn in the perspective formulas, and that's the same case even though it's drawn in the Fisher projection. So that just makes our life easier. Moving on, like this is a very simple sugar, glyceraldehyde, because it only has 3 carbon atoms. What if we're trying to determine the L or D, so whether like a complex sugar like glucose, that the one that we're looking at here is in its D isomer form or in its L isomer form? Well, we look at it, we determine it by the correspondence of its chiral carbon farthest from its carbonyl carbon with the configuration of glyceraldehyde. Well, this sentence is very long and complicated, so let's parse this. So the chiral carbon farthest from its carbonyl carbon. So first, let's identify the carbonyl carbon. By the way, let's just pretend that you don't have the answer given already. So we're trying to determine the isomer of this glucose molecule without knowing it. So let's first find the carbonyl carbon, which will be here, right? Because it's the carbon that's double bonded to an oxygen. And then we have to find the farthest chiral carbon. Is it this one? No, it's chiral, but it's not the farthest. Not this one either, not this one. It is this one. You might ask, why is it not this one? Well, keep in mind that for a carbon to be chiral, it has to be asymmetric, which means that it has to connect to 4 different groups. And here it's connected to 2 hydrogens, so it is therefore not chiral. So yeah, so we have this carbon that is chiral. And this is the farthest carbon from the carbonyl carbon. And the configuration of this one, it has the alcohol on the right, and that matches the D-glyceraldehydes configuration, right? So therefore it is a D-glucose. So similarly, if you want to determine this, the isomer of, so whether this molecule is a D or L isomer of glucose, you look at the chiral carbon farthest away from the carbonyl carbon, which will be this one, and then you'll see that the alcohol is facing the left, and that corresponds to the L-glyceraldehydes configuration, so therefore it is an L-glucose. Just a rule of thumb, most hexoses are D isomers. That's just, what nature decided them to be. I don't really know why, nor do biologists have like a, sort of uniform opinion on this. So yeah, but it's helpful to know that. Oh, by the way, later we'll see that uh, most proteins will have amino acids in the like D isomers of amino acids. So yeah. Moving on, like a complex molecule like glucose will have multiple chiral centers, right? So how do we know how many stereoisomers it has? Well, we have this formula. So if it has n number of chiral centers, then that means it has 2 to the power of n number of stereoisomers. So what does that mean? Well, for glyceraldehyde, remember that it has one chiral center, so n will be 1. And you plug in, n to 2 to the power of n, and that will be 2 to the power of 1, which equals 2, right? So for a molecule with one chiral center, it has 2 stereoisomers. But if you have a molecule with 2 chiral centers, then it will have 4 stereoisomers. Why? Because the first chiral center can assume an L or D position, configuration, sorry, and then the second chiral center can also have an L or D configuration. So therefore, you'll have 2 combinations, right? You can have LLDD, DLLD. So how many D isomers does aldohexose have? Take a moment to pause and think about it. And by the way, aldohexose is just a fancy way of saying glucose. So going back to our formula, we'll see that, we first figure out how many stereoisomers it has in total before we look at how many D isomers aldohexose has. So aldohexose has 1, 2, 3, 4. It has 4 chiral centers, right? So that means it has 2 to the power of n, which is 2 to the power of 4 stereoisomers, and that will be 16 stereoisomers. And out of that out of the 16 stereoisomers, half of them will be D isomers. So therefore, we will have 8 D isomers, and we'll also have 8 L isomers. Yup. Oh, sorry, my bad. Aldohexose doesn't mean glucose, all right? So aldohexose just means a carbon that has a monosaccharide that has a 6 carbon chain with an aldehyde group. And glucose happens to be one specific isomer of this aldohexose. And as we can see here, as I said before, there will be 8 D isomers. And so as we can see here, there will be 8 different names for the D isomers of aldohexose, right? So glucose is just a specific combination of the 4 chiral carbons configuration in this aldohexose. All right, so these are all aldohexoses, but they are the same molecule, though they are different isomers of each other. Why are they the same molecule? Well, they are, they connect to the same things, but the way in which they connect in 3D space is what determines what kind of isomers they are. So yeah, and then we look at the Fischer projection of glucose and mannose here, and we see that they only differ in one configuration, which is this top carbon, right? Because for the second, the third carbon here, it will have the alcohol on the left. That's the same case for mannose. For the fourth, it will have the alcohol on the right. That's the same case for mannose. It will have the alcohol on the right for the fifth, and that's the same case for the mannose. Only the first one differs in the configuration, right? So we call 2 isomers that differs in one configuration out of all of them epimers. And it is one and only one difference in configuration, right? So if you have, let's just compare glucose to, let's say, altrose, right? This carbon and this carbon, they have different configurations compared with this carbon of glucose. So therefore, they are not epimers because they have 2 different chiral centers. Epimers needs to only have one different pyrocenter. So we can say that mannose and glucose are epimers. Okay, so here's your job. Find another pair of epimers. All right. Hopefully you found it, and if you have any questions, you can ask me during our real-time online session. Yeah, and then I'll move on. So just to recap, L-glucose and D-glucose are enantiomers of each other. They are mirror images of each other. And D-glucose and D-mannose are epimers of each other, right? Because they are they differ in only one chirocenter. And D-mannose and D-idose are stereoisomers. So D-idose will be here, and D-mannose will be here. And L-glucose and D-idose will also just be stereoisomers of each other. And the four of them are all stereoisomers of each other. All right, so epimers and enantiomers are, how do I say it? Like specific cases or special stereoisomers. All right, so if a molecule and another molecule, sorry, if two configurations are enantiomers of each other, they must be isomers, like stereoisomers of each other. But if they are stereoisomers of each other, that doesn't mean that they have to be enantiomers of each other. The same case goes for epimers. So let's take a moment to pause. This is a lot of information. All right, take a deep breath and we'll move on. Oops. Okay, so common monosaccharides have cyclical structures. So, the molecules that we've been looking at before, they are drawn in this linear structure, but turns out they go through a reaction. For aldehydes, they react with an alcohol and produces an hemiacetal. All right, they produce a hemiacetal. And for ketone, they react with alcohol to produce a hemiketo. And then for hemiacetal and hemiketo, they can also react with another alcohol to produce acetal and keto, respectively. So, if we look at the glucose here, it turns out that under, our physiological conditions, it will have, it will favor cyclical structures. So it turns out that the alcohol here comes from, the same molecule, right? So the alcohol attacks the aldehyde carbon, and then it forms two different, once again, two different isomers of each other. So it forms an additional like stereoisomeric property due to the cyclical structure, because the way in which the alcohol attacks the aldehyde affects the stereo outcome, right? So if we examine the organic chemical mechanism of this process, we'll see that due to the free rotation of the single bond here, when the alcohol attacks, the oxygen could be the one that's facing up, or the hydrogen could be the one that's facing up, leading to ultimately different products, right? You can have the beta D-glucopyranose, which has the alcohol group facing up, or the alpha D-glucosepyranose, which has the alcohol facing downwards. And, this carbon will, have different configurations compared with this carbon. So then we call the isomeric forms of monosaccharides that differ only in their configuration about the hemiacetal or hemiketocarbon atom are called anomers, right? So alpha and beta means that they are anomers of each other, while D or L means that they are enantiomers of each other. So, we add that to our beautiful graph of stereoisomerism. All right, so you might have noticed that we were calling the ring glucose or glucose in the ring form glucopyranose. Like, why is that the case? Because the ring form of the glucose resembles this molecule called purine. So that is why we call it glucopyranose. Of course, we need this prefix, right? Alpha or D, beta or L or something like to know its, to know more about it. And then for fructose, because it looks like furan in its ring structure, it's called fructofuranose. So a general rule of thumb is that, if they end up with a 5-membered ring, they're likely to be furanoses because they resemble furan. And if the linear structure forms a 6-membered ring in its cyclical form, then it probably looks like puran and it will have puranose in its name. All right, then we'll look at how do we convert Fisher projection into, this 3D cyclical structure, which we will call the Haworth perspective. So the rule is that the groups on the right, they will go down. So as you can see here, I highlighted them as blue, and they will end up below the plane of this ring. And the one that is on, the groups that are on the left will end up upwards, right? So they're highlighted in pink. And then for, how do we determine whether it's a D or L isomer? So if the 6th carbon is up, then that will be a D enantiomer. If the 6th carbon is down, that will be an L enantiomer. All right. And then also, how do we determine its, anomericism? Well, if the C1 carbons hydroxy group, or in other words, alcohol, or in other words, OH group, is opposite to the C6 carbons, then we will call it alpha. If the OH group is upwards, and it is at the, the 6 carbon is also upwards, that means they're on the same side, we will call it, a beta anomer. Take a moment to pause again and take a deep breath. All right, let's move on. We only have a bit more to go. So just to complicate things further, they are actually not planar hexagons in real life. So they're not literally like a hexagon. They also have a complex 3D structure, and we call this conformation the chair conformation. The other one will be boat conformation. And the reason for this is because, the groups might have steric hindrance. And to minimize that, they want to adopt this conformation so that they are as far away from each other as possible. And this structural notion determines the properties of some polysaccharides, which we're going to look at in the future sessions. Just keep that in mind. All right, just to complicate things up even more, other groups can also attach to the monosaccharides. So, we won't go through all of them, but I'll point to some of them. For example, you can have, this this group with a nitrogen, and also some kind of like a carbonyl or stuff like that. If this group is attached to the second carbon, it forms this thing called N-acetyl beta D-glucosamine, and it can form structural polymers, and it exists in bacterial cell walls. And for here, you can have deoxy sugars. So deoxy sugars means that the sixth carbon won't have any oxygen, right? As other sugars, other sugars, they have oxygens on the sixth carbon. So deoxy sugars don't. And for example, beta L fructose, fucose, or fructose, I don't know. They can like be in glycoproteins and glycolipids. Alpha L rhamnose, they can be in plant polysaccharides. And then moving on, you can have acidic sugars, with this carboxylate group. Yeah, and they can also be in glycoproteins and glycolipids, typically on animal cell surfaces. All right, another kind of sugar we're looking at will be sugar with phosphates. So just to remind you, you've actually seen that before if you have taken the AP Bio course. So some of its function is that it traps the sugar. So by attaching the sugar to a phosphate group, it traps the sugar within a space, for example, a mitochondria, since most cells don't have membrane transporters for phosphorylated sugars, so the sugar won't be able to move across the membrane. Also, it activates sugar for subsequent chemical transformations, for example, in cell respiration due to that charge, which really destabilizes it, and we'll examine it further when we come to this unit. The third function is that sugar phosphates are actually components of nucleotides, right? So remember that a nucleotide is composed of a phosphate connected to sugar connected to a nitrogenous base. So if you see this sugar as connected to the phosphate, which becomes a sugar phosphate, then sugar phosphates are a part of nucleotides. So yeah, these are the significance of the sugar phosphates. All right, and this will be the end. Thank you all for watching this video. I hope you enjoyed it.